Thank you. Good morning, everyone. I am really delighted to be here, especially because I was part of the group from 2012 that tried to tackle this question. And we are back here all over again, and I suspect that we are going to be struggling with some of the big picture ideas as much today as we did in 2012. But what I want to do in the time that I have is do a quick review of what some of the conclusions were from this working group from 2012, and just give you a few of my thoughts. These are going to be big picture thoughts about where we are, where we've made progress, where the field seems to have stalled, and then maybe just raise a couple of questions for consideration as we go through the day. So as this was already introduced, this was a tremendous opportunity. It was a phenomenal set of people who came together to really ask whether or not, back in 2012, we could come up with a definition for healthy human gut microbiome. And what I've captured here on the bottom of the slide here is, we don't have a pointer, but we probably don't need one. I think this was the concluding sentence from this paper. Elucidation of the properties of healthy microbiota would provide a target for dietary interventions and microbial modifications aimed at sustaining health in generally healthy populations and improve the health status of people exhibiting disrupted microbiota and associated diseases. And this is a little bit like mom and apple pie. I don't think anybody would argue that this is a really laudable goal. But I think as we are going to see today, as we did then, the devil is in the details. I tried to pull out from that paper some of the important concepts that the group agreed on. Resistance and resilience are key features of a healthy microbiome. A healthy microbiome cannot be defined by an idealized community composition. A healthy set of metabolic functions needs to be defined. Certain microbial distributions may increase susceptibility to infection or disease. We cannot distinguish between dysbiosis as a cause or consequence of disease. The concept of a core microbiome defined by taxonomic data is compelling. I should have probably noted the but squishy was not actually in the paper, but this was more how it was presented and I think what we felt. And the concept of enterotypes in the gut should be further explored. And I would argue that perhaps with the exception of the bottom 2 points on this slide, we haven't really made much progress. And I think as you saw in the just the opening comments, a lot of these ideas have never died. They're still with us, and we're still struggling with these definitions. Back in 2012, the idea that you could use taxonomic data to define a core microbiome was certainly circulating, and I suppose it's a matter of how you define this. If you define this at the phylum level, well, yes, of course, there's a core microbiome, but that's really rather meaningless. And if you get down to genus and species, and you look very, very broadly across multiple populations, while we might be able to identify a core set of organisms, it's an ever diminishing set of organisms. And it's actually interesting when I look back that the concept of enterotypes was sort of front and center back in 2012, but we've had the benefit of a lot of additional studies, and I think that concept died many years ago. But a couple of comments I guess I just want to make, and it should be noted, I think, that what we were dealing with back in 2012 was by and large mostly 16S ribosomal RNA gene sequence data. We still deal with an awful lot of that today, and even with that, there have been some interesting observations, and I think one of these, whether ethnicity or other host factors, have been really shown to associate with variation in microbial abundance when we use 16S as a metric. And I just want to touch on a couple of papers that were published this year because I thought they were really very interesting for a couple of different reasons. This is, and I actually happened to be one of the reviewers on both of these papers and was really excited when I saw them and was hoping that they would be published, and they were. And this is the first one here depicting the composition of gut microbiota in a population with varied ethnic origins but shared geography. This paper described what was called the Helius Study. And this study looked at a group of individuals all living in Amsterdam that self-assigned ethnicity, and the groups were divided into different ethnic populations. I'm not going to go through them here. Dutch, Ghana, Morocco, African, Surinamese, South Asian Surinamese, and Turkish. These were relatively recent immigrants to Amsterdam, so we weren't looking at multi-generational effects. And I think the take-home message here from this paper was that when factoring out socio-demographic, lifestyle, or dietary factors, there was still a segregation of the microbiota based on 16S data that seemed to track with ancestry or ethnic origin. This is perhaps not surprising, but I think what was well done about this study was that it was enrolling people from a given geographic area and looking at a host factor. And it certainly raised the question to me, and I think it's something that we will debate a fair amount today. Do we pay enough attention to host factors when we are studying the microbiota? I'm not sure we do, but that's a tough nut to crack in terms of what to really look at. This was another paper that really captured me, also published in Nature Medicine, the title, Regional Variation Limits Applications of Healthy Gut Microbiome Reference Ranges and Disease Models. And this may be even more applicable to what we're going to be talking about today. And this was a study that enrolled, if I recall correctly, almost 8,000 individuals from Guangdong province in China. And the province was then divided into regions and into neighborhoods. And long story short here, a tremendous amount of metadata were collected. I guess the pointer here doesn't work. But if you look at panel B on the top right, these were all the metadata that were looked at. And what was very clear was that it was districts, it was geographic location that was the primary driver of the differences that were observed here in, if you look in panel C, if you look at a PCA plot separating these various populations. Obviously, geography like this is an umbrella for a lot of factors. It could include ancestry, as we saw in the previous study, other lifestyle factors, but this by and large was the primary driver. And what I also found to be equally interesting and perhaps just as important in this study was what happened when the investigators went into a particular region, identified individuals with one of 4 diseases, type 2 diabetes, metabolic syndrome, obesity, or fatty liver, put together a disease cohort, redid the 16S analysis, and came up with a set of taxa and changes in relative abundance that were associated with the disease in that particular region. And I think what is so telling, and I think what we've come to appreciate from looking at many, many papers that have been published, is that if you take those training sets of data and then go somewhere else and ask, does this microbiota profile identify patients in another region with those same disorders? The answer was no. And what these dot plots here are showing you is in blue the areas under the curve for the training sets, if you will, and then the dots in pink, the areas under the curve when those community compositions were applied in a different region. So again, I think what this is telling us is that host factors make an enormous difference. And I don't think this is anything new, because we know that we struggle with inter-individual variation in the microbiota as defined by 16S profiles. But I think these two papers that were published this year, perhaps even more so than some of the work that has come out of the Human Microbiome Project, really drove some of these learnings home. I'm not going to spend any more than just a couple seconds on this slide. This is also from this paper about geographic variability. And the point here, this is looking at a difficulty gradient that could be used to interpolate and extrapolate the MET-S model. And the point is here, if you look again at areas under the curve, these predictions, these models seem to work well in a very small geographic area, but the utility of these tend to disappear as you move to look at other populations. So I think that this idea is something we will be struggling with quite a bit today in our discussions. I just want to make a couple of other comments about starting to move away from 16S metrics to begin to look at function, because I think even going back to the 2012 paper, there was consensus that we needed to define the appropriate functional metrics in order to define a healthy microbiome. And this is a paper that my lab published earlier this year. And what I want to focus on here are the functional differences that we identified in this study. And this was an interesting study. We were looking at, this was a population of human subjects recruited in the UK, in Oxford, by Andy Pollard and colleagues. And this was actually a human challenge trial with wild-type salmonella and a salmonella vaccine. The vaccine issue of this really doesn't matter today, but what we did was really move beyond 16S and begin to try and characterize the communities and individuals based on function, and we were using metatranscriptome analysis. And what we found in the 30 patients, which granted was a very small subset, but what we found was two different transcriptome community types. The relative abundance of transcripts from different taxa are shown there in the lower left, and we saw two community types, one dominated by a very high percentage of methanogen transcripts shown in green on the top, and the other community type was dominated by transcripts from a diverse collection of firmicutes. There were differences in the gene content in these two communities based on our analysis, and we also saw differences in a number of functional categories shown here in D, and I realize that you can't read that, but the point that I want to make is that there were differences. One of the big differences in the methanogen community was that we found all the genes involved in the methanogenesis pathway. And this suggests that there may be some interesting differences here, because methanogenesis is one of three mechanisms known to be involved in disposal of hydrogen that's generated during anaerobic fermentation in the colon. So it suggested that perhaps these individuals were disposing of hydrogen differently, and because we're able to identify various networks of interacting taxa, it suggests that these functional differences may translate across various individuals. But we also acknowledge that our understanding of this process of using hydrogen in the gut and where it takes place is somewhat fragmented. But you might ask, well, so why does this even matter? I mean, okay, so what? You found differences in functional profiles. Couple of things. These differences in functional profiles did not map in any way back to the 16S data sets that we generated. So this was a completely different view of the microbiota in these individuals. And what we did also see was that in terms of the ability to be susceptible or not to developing typhoid disease after challenge with wild-type salmonella, it really did matter. And in the community dominated by the methanogens, the clear majority of individuals did not develop any clinical symptoms, irrespective of whether or not they received any vaccine. So here was an observation that was unexpected, and one might ask, does this matter in terms of health? You might say that it does. And so here we saw a functional profile based on metatranscriptomics that suggested there was indeed a specific outcome of all of that. But we have a long way to go from these kinds of observations to mechanisms, and I think that's what we all struggle with, particularly in studies with human subjects. So I think ideally where we would like to end up is to be able to develop some sort of common approach to clinical test development that would allow us to use those data to say that we are looking at a healthy microbiome. Ideally, and this is probably too idealistic for what we're talking about here, would be something like what we are all familiar with in terms of cholesterol measurements. You make a measurement, you can make it across all populations, and we have come to believe that depending upon which category you fall into, you are either healthy or in an ideal range or not. But I think what we have to ask ourselves with regard to microbiome measurements is what should be measured, can appropriate target ranges even be defined, and also how practical are these measurements to make. It would be wonderful if we could think about doing something with a blood sample, but I've seen very few microbiome functional studies that are making measurements in blood. And if you require a fecal sample, then while that's certainly doable, it becomes a little bit more cumbersome. And I just want to conclude with one example here, which has already been mentioned, short-chain fatty acids, and in particular butyrate. I suspect that there would be agreement here that butyrate is a beneficial metabolite from the microbiota. It is an energy source for colonocytes. About 70% of the butyrate that is produced is taken up by the colonocytes. Butyrate as a molecule has a very short half-life, both in the intestine and in the blood. So that already presents some challenges. Butyrate inhibits histone deacetylate. It stimulates Treg cells in the colon, and it polarizes M2 macrophages. These are all thought to be important outcomes, very much focused on the impact of butyrate on immune function. Is that something we should be measuring? Maybe we have to measure it in the colon, though, and then all of a sudden that becomes really not doable. We know that short-chain fatty acids bind to a couple of G-protein coupled receptors in the epithelial cells in the gut, and they regulate any energy metabolism by releasing gut hormones GLP-1 and peptide YY. Not practical to measure those in the GI tract, but might we be able to measure the distal effects that are known to take place in the liver and in brown and white adipose tissue? And finally, short-chain fatty acids impact intrinsic enteric neurons, and autonomic intestinal innervation modulates the gut-brain communication. Is this something we should be looking for? Could we be looking for signaling molecules in the serum? These are all open-ended questions. I'm not even going to attempt to provide an answer. But the point of this really more is that here are examples of a metabolite that I think we would all agree is important. And yet, I think we struggle with what do we measure. So I would then put forward for consideration for some of our discussion today some questions. Should we measure specific microbiota metabolites? Or instead, should we measure host factors that are impacted by these metabolites, or should we measure both? How many microbiome and/or host variables will be necessary to capture a meaningful biosignature of gut health? What experiments are necessary to understand how these to-be-determined microbiome parameters vary across populations and in response to various perturbations, diet being the most obvious one? And will a single reference range of whatever marker we think might be usable, is a single reference range sufficient? Or going back to some of my earlier comments, do we need to consider some of these in a population-specific manner? And finally, another idea just to put out there for consideration. So much of what we have studied and measured with regard to the gut microbiome, whether it be 16S ribosomal RNA gene data, whether it be functional data, transcriptome, metabolome, has been derived from studies of individuals in developed countries, by and large. And we know from some of the studies that have been published, and what I'm referencing here is work from Maria Gloria Dominguez-Bello, that if you look at microbiome composition using 16S data in some of these isolated populations, and she's done a lot with various Amazonian tribes, what you see is that the composition of the gut microbiome in populations that don't eat a Western diet, have not had the same kind of exposure to antibiotics that we all have, are very, very different. And she and Marty Blazer and others have even suggested that perhaps we need to rethink our definition of a diverse microbiome, because if we're defining it based on what's present in all of us, we may be missing an even bigger picture. So what we may think is a healthy microbiome may indeed be a suboptimal microbiome, and that just throws another monkey wrench into all of the discussion. So I just want to finish with some acknowledgments, some folks from my lab who were involved in the study that I mentioned, but people who aren't listed on this acknowledgement slide are all of you in the audience, and really everybody in the entire field who has contributed to our knowledge up until this point. And I think where we are in some respects is still a little bit like that parable of the blind men and the elephant. We are measuring, we are looking at a lot of different things, and we don't yet have the entire picture. And so hopefully today what we can at the very least do is put together a set of recommendations for work that needs to be done that may not get us to answering the question about quantifiable metrics, but at least get us a few steps closer to collecting the data. Thank you.